field directly, and I wish I could say for the patients that have mitochondrial diseases, the poor children, that they could take a pill or a poly pill to get rid of the bad symptoms that go with these mitochondrial diseases. So today I'm going to tell you about a pathway that we think could be used for a therapeutic approach to potentially treat mitochondrial DNA diseases caused by mutations in the mitochondrial genome. And so, if we look at the mitochondrial genome, it is a 16 kilobase circle, and it lacks introns and contains 13 genes that end up in the membrane for OXPHOS. It has a displacement loop, or D loop, which controls replication, and what happens is that there's two large transcripts that are synthesized, and they are going to be separated by tRNAs, and this is called the tRNA punctuation model for transcription. What makes mitochondrial diseases difficult to treat is that when we look within a cell, a single cell can have anywhere from, let's say, 60 to 600 to an oocyte with up to 10,000 copies of the mitochondrial genome. So we can't do simple gene therapy. In addition, we aren't able to easily transform a mitochondrion. So the alternative therapy that we've identified is through this unexpected new pathway that allows RNAs to be imported. And so the story started with a collaboration with Mike Teitel, a pathologist at UCLA, and he was working on TCL1 for T-cell lymphomas. And what he thought is that there must be some other interacting proteins that would work with TCL1 to help modify this cancer. So he took what we would normally do, a good biochemical approach, and he pulled down one protein that interacted. And this protein was an 85 kilodalton protein, and it was identified by mass spec to be a single partner protein called PNPase. And even though he had a new interacting partner, it still wasn't clear what this protein might be doing. PNPase is, by homology, an RNA degrading enzyme. In prokaryotes, it's part of the RNA degradosome. And if we look, we see that there are homologs in chloroplasts and plant mitochondria. But when we go over to the animal world, we find that it's absent in fungi and trypanosomes, but it shows up in worms, fly, and vertebrates. Interestingly, we still don't know what the significance is of the interaction with TCL1 and whether it's real, but this gene is induced by type 1 interferons, which is related to T-cell lymphomas. In addition, it's also upregulated in some cancers, and some early studies in the protein suggested that this protein was localized in the cytosol. So we started out to characterize the enzyme, and we predicted that it would degrade RNA. And what we can show is that PNPase is able to degrade RNA. However, the RNA must have a poly A tail. So it seems to be dependent on the presence of poly A tail for degradation of RNA. So then when we started looking at the protein in more detail, when I looked at the N-terminal region of this protein, I saw that it had a canonical N-terminal targeting sequence. And for proteins that are destined for the mitochondria, a great number of them will contain an N-terminal targeting sequence that contains positive charges which are present on one side of the amphipathic helix and hydrophobic charges which are present on the others. So this suggested that PNPase was probably a mitochondrial protein. And so we started some biochemical approaches to verify that that's where the protein was localized. And what's nice is we can take advantage of biochemistry, and from work we've been able to show, we know that protein import is highly conserved from yeast to animals. And so we can take yeast mitochondria as a model, and what we did in our first step is we made radio-labeled PNPase. So here we have PNPase that is radio-labeled, and then we can incubate it with mitochondria, and over time it becomes imported. However, import is not so efficient. It was not so efficient, and so what we do is we add protease. What protease does is it will remove all of the precursor that is not imported. And import depends upon the presence of a membrane potential. And what we were able to show is that PNPase is actually imported into mitochondria, and this N-terminal targeting sequence is cleaved. So this verifies that it is indeed a bona fide mitochondrial import, mitochondrial protein, and not a cytosolic protein as has been suggested in the literature. We also included additional controls, SU9-DHFR, which goes to the matrix, and is also processed upon import into the mitochondria. So at this point, I predicted that PNPase would be in the mitochondrial matrix. It had been suggested as having a role in RNA degradation, and this is the compartment that we find RNA. However, we did the next step, which was to localize it to the matrix or the intermembrane space. And for doing this, we use a method called osmotic shock. What we do is we lower the osmotic concentration of the mitochondria, and then the outer membrane will lyse because the inner membrane expands. And when we do this in the presence of proteins which are in the matrix will be protected, whereas proteins that are in the intermembrane space will be degraded. And so if we look at this, here we have HSP60, a protein which is found in the mitochondrial matrix. As we lower the concentration of the osmoticum, we find that HSP60 is protected from protease. And then when we look at TIM13, a protein which is present in the intermembrane space, we see that it becomes degraded. And then with PNPase, if we didn't add protease, we saw that it remained associated with the membrane fractions. But upon the addition of protease, we saw that it was extremely sensitive. So we have a new protein that's involved in RNA degradation in the intermembrane space. At this point, it doesn't really make sense. So we have a good location, but we were still somewhat suspect. So we moved into a more detailed analysis to try to figure out how this protein was imported into mitochondria. And this is really important when you want to start dissecting new import pathways because you really need to know where a protein is. And so we again turned to yeast, and we made some mutations in some of the key enzymes that might be involved in processing and importing the protein. And so, as you can see in wild type, here we have the mature form of PNPase, which has been imported into mitochondria. And then we have a mutation in MAS1, which is this matrix processing peptidase, which cleaves the N-terminal targeting sequence. And we can see here that PNPase is no longer imported. So this indicates that we need the matrix processing peptidase for import. And then we thought that an intermembrane protease, IMP1 and IMP2, might play a role, but we found that there was no effect on the import of PNPase. But finally, when we deleted another protease, YME1, we saw that PNPase was again not imported. Now this is somewhat of a surprising role. YME1 is thought to be a general protease that degrades proteins that are misfolded. We didn't really expect it to have a role in the assembly of PNPase. So we wanted to verify by a couple more of experiments to show that YME1 specifically plays a role in PNPase import. And what we can do is we can knock YME1 out, and then perform these same import assays as I have shown you, and we can see clearly that the lack of YME1 results in a defect in the import of PNPase. And to further confirm it, we can do import and cross-linking experiments, and what we should be able to show is that PNPase will be bound by components that mediate its import. So we are able to import PNPase, and then we add chemical cross-linkers followed by immunoprecipitation. And we see that these import components, YME1, TIM23, and TOM40 bind specifically to PNPase, but cytochrome B2 does not. So we can summarize this as showing that we have a new protein in the mitochondria, and it's assembled via a new import pathway. It comes in via the TOM and the TIM translocons, and then the matrix processing peptidase cleaves it, and then YME1 actually pulls it into the intermembrane space where it assembles as a complex. Of course, we still have a problem. We don't know what the specific function of this protein is. So we came back to some biochemical approaches, and we knocked down PNPase by using RNAi. And what we found is what we find in a lot of typical mitochondrial stories. We found that there was a decrease in mitochondrial respiration. We had decrease in the activities of the mitochondrial respiration complexes, And we also found that there was a problem with assembly of complex 5. So we knocked down PNPase, and we have an impairment in respiratory function. This also occurs with the shift in metabolism. If we look at how the cells are growing, in the cells that we've knocked down PNPase by RNAi, we find that their media becomes more yellow, and this corresponds with an increased abundance of lactate in the media. We also find that the ATP abundance is less, and in general we see that the cells do not die, but they just slow down in growth and fail to proliferate. So at this point, what we can see when we have a decrease in PNPase in mitochondria is that we have a change in the ATP to AMP ratio. We have an increase in glycolysis and lactate. And we also find that there's an increase in the phosphorylated forms of AMP kinase and a decrease in cell proliferation. So this is really not surprising. And at this point, it's somewhat disappointing. It's hard to publish papers if you don't know exactly what proteins do. So at this point, a postdoc in my lab, Gang Wang, and a graduate student in Mike's lab, Eriko Shimada, came back with a new approach and focusing more on biochemistry again in the mitochondria. We also had a mouse model that we could take advantage of, and that really helped break through and help us understand how this new pathway works. And so one of the first things we did was looked at how PNPase assembled. And the reason to do this is because we want to work in yeast, because we can do a lot of genetics and biochemistry, but we also want to work in mouse. And what we find is that in either system, the cells assemble, that the PNPase assembles in the same size complex. So we didn't think that there would be some obvious partner proteins that PNPase would have. So we looked anyway for partner proteins, and we had PNPase, we had a tag on it, and we did pull-down experiments. We got PNPase back as you'd expect. We also got back some lower bands, but mass spec showed that they were just degradation products. So again, PNPase seems to be a lone protein working on its own. We knocked it out in mice, hoping to have some answers. PNPase was essential for viability, and the mice died at day 7 or day 8, which is not untypical for mitochondrial proteins. We were, however, able to get a knockout in which PNPase was knocked out in the liver. And we find here that the mitochondrial have an aberrant morphology. The cristae are not as well developed. And in general, there's a decrease in respiration by about 50%. However, the mice are able to survive, which is somewhat surprising. So we started looking at mitochondrial function, and here we found that there was a general translation defect. Translation was decreased by about 50 to 60% in these mitochondria. And so that led us to a question: if translation was defective, perhaps RNA processing is. And as I told you, we have this tRNA punctuation model. And what happens is, for example, with COX-1 and COX-2, the two tRNAs will fold, and these will become sources for the processing enzymes to come in. And then we'll be left with mature transcripts for COX-1 and COX-2. And so we reasoned that if we had a defect in processing, that we might find longer transcripts. So we used RT-PCR, and we developed primers that looked just at COX-1 or looked across the COX-1-COX-2 junction. And what we found in our knockout mice that lacked PNPase was that these longer RNA transcripts were occurring. And when we did northern blot analysis, we found the same result. Once again, there was an increase in longer forms of unprocessed transcripts. So this would explain the overall general defect in translation. So then we started considering, how does an enzyme in the intermembrane space regulate RNA processing in the mitochondrial matrix? Well, we've known for some time that there are RNAs that are imported into mitochondria. Every organism has at least one tRNA that's imported. The 5S ribosomal RNA is imported. The RNAs for RNase P and MRP1 are also imported. These are required for mitochondrial DNA replication and transcription. There's also an exciting field developing right now that show that microRNAs are imported into mitochondria and might play a specific role in attenuating mitochondrial gene expression. And finally, there are viral RNAs that target complex 1. So there's a lot of RNAs that are moving into mitochondria. but nothing is really known about how these RNAs are imported. And so we thought that perhaps PNPase, given its ability to bind and degrade RNA, might be the first component in this type of novel pathway. So we began studies to look at a potential role for PNPase in RNA import. And as you can see here, we used RT-PCR to look at the levels of RNase P RNA, and we found that the levels were much higher in mitochondria isolated from our wild-type mice from the livers. In contrast, when we isolated mitochondria from the knockout liver mitochondria, we found that the levels were much lower. So if the levels of RNase P RNA are lower in mitochondria, we would predict that PNPase should be able to bind to it. So we looked at these same RNA import assays, and then we did pull downs with PNPase from our tagged cell lines, and we were able to show that RNase P specifically bound to PNPase. To give you an idea of specificity, when we look at COX-1, which is in the mitochondrial matrix, we see no interaction. And we look at GAPDH, which is in the cytosol, and again we see no interaction. So this shows that PNPase specifically binds to RNase P, which is being imported. We also looked at processing of tRNAs, and what we can show is that in mice that lack PNPase, that we actually have a decrease in this tRNA processing. So the RNase P is important then for processing of the tRNAs. So we wanted to once again show that this was specific, and we moved back into yeast to develop an in vitro import assay. And what we can do is we can take RNA, an RNase P RNA, add it to mitochondria, and then look over time and see what is imported. And if we look at our cells in which we have PNPase overexpressed, we see that there's an increase in the RNase P RNA. And then moving into mammalian mitochondria, we see the same thing. If we downregulate PNPase by RNAi and compare the import of RNase P, we see that it is increased in the wild-type cells. In addition, we see the same things with MRP RNA, the 5S and the 5S ribosomal RNA. But again, GAPDH, which is present in the cytosol, is not important. So at this point, we've identified a new component that mediates RNA import. What does this have to do with potentially correcting RNA mutations in the mitochondrial genome? Well, during this, as we started to figure out how some of these RNAs were imported, we identified a specific stem loop region in RNase P and MRP that was important for its import into mitochondria. So we asked the question, can we append this to another RNA and mediate import? So what we did is we took this stem loop region and we attached it to GAPDH RNA, and then we performed import assays. And what we were able to show is that when we have this targeting sequence attached to the GAPDH RNA, we are able to import this cytosolic RNA into mitochondria. This was a pretty exciting result because nobody really understood how RNAs were imported. So then we started asking more detailed questions. Could we actually import a corrected RNA back into mitochondria? And so what we did is we took the COX-2 the COX-2 gene, again appended it to this targeting sequence, and we were able to actually import it into mitochondria. And as a result, we could see that the protein was subsequently translated. So we were able to append the import sequence, followed by importing this RNA. It was translation competent, and it was able to be translated into the mitochondrial genome. So can we use this to potentially treat mitochondrial diseases? According to the United Mitochondrial Disease Foundation, about 1 in 5,000 children has a mutation in the mitochondrial genome and has a mitochondrial disease. A lot of these are really tragic diseases. Children die at a young age, and they have a variety of symptoms. There will be deafness, blindness, they'll have problems with movement. In addition, as we age, There's also a suggestion that we accumulate mutations in our mitochondrial DNA genome that could also be result in our inability to produce as much ATP. And if we start thinking about our stem cell therapies with the goal of taking adult stem cells and using them as a regenerative medicine for us, we'll have to consider what the mitochondrial DNA genome looks like. If we want to try to treat some of these diseases, for example, we can think of these neurodegenerative diseases such as Parkinson's, we have to consider what the mitochondrial DNA genotype is. Because if we take these cells from adults and they already have a compromised mitochondrial genome, we might not have enough energy in this therapy to really improve the disease. So a possibility is that this approach might be useful in some of these new types of mitochondrial, in some of these new types of therapies that are being developed. So we took two different cell lines. They were hybrid cell lines. One has a mutation, one tRNA that results in May loss, and another one has mutations in mitochondrial tRNA that results in MRF. And again, both of these are tragic childhood illnesses. The children typically don't live past the age of 10 to 12 years. And so one of the first things we wanted to do was in vivo, before we moved in vivo, was to show that we could actually import these tRNAs in vitro. So we took this tRNA and hooked it to the targeting sequence that would direct its import. And as you can see, in mitochondria isolated from wild-type mice, we're able to import this tRNA fine, but in the mitochondria that do not have PNPase, we have a block in import. So the next thing we wanted to do was see if we could actually rescue translation in the mitochondria. And in this case, what we find is when we import these corrective tRNAs, we're able to slightly rescue the rate of translation. So we wanted to move then into cultured cells. We wanted to make stable cell lines that express these tRNAs and see if we could rescue it in a more in vivo setting. And our first attempts were not successful. And upon further analysis, what we wanted to do was we wanted to take this tRNA with its targeting sequence. It would be exported out of the nucleus into the cytosol, and then it would have to find the mitochondria. And from the early analysis, we realized that what was happening was it was just ending up in the cytosol, being processed as if it was a normal tRNA. So we started manipulating the tRNA export pathway, and we found that if we changed some of the base pairing in the stem region, we could bypass the canonical processing. So we made some mutations there that allowed us to get this corrective tRNA from the nucleus into the cytosol. But we still didn't have luck. We weren't able to get this tRNA to the mitochondria. So some nice work that's been done in the field has shown that prokaryotic RNAs actually have at the 3' an untranslated region which acts as a signal to take them to mitochondria. So we decided we should take this 3' UTR and append it onto the tRNA and see if that would actually direct our tRNA now from in the cytosol to the mitochondria. And when we did this, we were able to finally rescue translation. So if we take tRNA precursors with all 3 elements, the targeting sequence, the 3' UTR, and the alteration in the stem loop region, we are able to restore translation in these cell lines. And if we look on a blot analysis with an inorganello translation, here we see the control cell lines. We have essentially no translation occurring. And when we add our corrective tRNAs, we're able to increase it by about 75%. And this results in the presence of COX-2 and ND6, which are now detectable by immunoblot analysis. So what we're currently trying to do at this point is we're trying to expand this and see if we can target other types of corrective RNAs. There's examples in which we have mutations in cytochrome oxidase and complex 1, and these are some of the current targets we're working with. So by using a biochemical approach and a different type of talk than you're used to, we've been able to show that we have actually a new pathway to import RNAs into mitochondria. PNPase is an RNA binding protein, And it also has an ability to degrade RNAs. So we think that it acts as a gatekeeper. So RNAs that come in that shouldn't be imported are probably degraded, but RNAs that should be imported will then pass through into the mitochondrial matrix. Some of the obvious questions that we're trying to address right now are, how do these RNAs get across membranes? This is still a big question in the field. Some of our preliminary analysis suggests that porin or VDAC on the outer membrane might be one of the components. In addition, we're working in a more collaborative effort. We're trying to now look at targeting corrective RNAs into mitochondria. We have a collaboration with Doug Wallace. We're trying to target corrective RNAs in a mouse model that has a mutation in mitochondrial DNA. And in addition, we're also looking at this as a possible approach in stem cells to see if we can target corrective RNAs in adult stem cells should they have mutations in the mitochondrial genome. So with that, I'd like to thank you for your attention and acknowledge the people that have done the work. This is a collaboration with Mike Teitel. In his lab, Xiaowen Chen, Sam French, and Eriko Shimada have done the work. We've had an ongoing collaboration with Bob Lightollers to look at some of the RNA import pathway. Carlos Morais provided the cell lines, and then Ivan Tarasov and Nina Antelis in Strasbourg have been helpful in collaborations in discussing this pathway.